Hello friend, how are you? All are welcome to my channel GetEasyMedicalMicro. Today I am discussing about the bacterial growth curve. When a bacteria, single number of bacteria introduced in any culture media, that time what will happen? Okay, it has 4 steps. What are that? The step 1 is the lag phase, log phase, stationary and decline phase. Okay, so what are the 4 steps? Why you divided 4 separate phases? Okay, so the all step we are present graphically as per time duration. Okay, here we seen the number of cell, how much cell increase. Okay, and another one in respect to time. Okay, now comes to the lag phase. Okay, in that phase, when a single number of bacteria introduce any suitable culture media. Okay, again I am telling the a single number of cell introduced in a suitable culture media. That time what will be happened? That time it never takes rapid growth. It never takes rapidly growth. Why? What is the reason? It takes few time for the adaptation. Okay. So generally when this is the, suppose this one is the culture media. Okay. I introduce a bacteria. Okay, like this. I introduce a bacteria. Okay, culture a bacteria. So that time what will be happened? What type of facility generally given by this media? Number one is proper nutrition, present. Okay, number 2, pH is normal. Number 3, no accumulation of toxic product. Number 4, O2 supply. Number 5, number of cell. Okay, we these 5 suitable condition present in this media. So at first proper nutrition present in that media. Normal pH, no pH is changed, not acid or not acidic or alkaline. Another one is the no accumulation of toxic product. Still bacteria never growth, so bacteria never accumulate any toxic material. Okay, not release any toxic material. When bacteria release toxic material, they accumulate inside the cell. Okay, so that time no accumulation of toxic product. Next, O2 supply. O2 supply is still sufficient. Okay, number of cell growth. Number of cell growth nil. So all are favorable condition present, but it never takes growth. Why? Because it takes, after introduce a colony on suitable media, it takes few time for the adaptation of the nature. Okay, it release some enzyme. Okay, so it have to adapt the total nature. Okay, the media nature and all about things. So that phase is called the lag phase. So in that lag phase, bacteria will never grow. It will take some time to adapt the nature of the media. So here we see, as per respect to time, according to the growth factor, you can say the growth factor. Okay, so this one is lag phase. No growth is seen, so this one is lag phase. Okay, number 2, log phase. So what will happen in log phase? Bacteria will start grow. Okay, bacteria will start grow because all favorable condition is present inside the medium. So nutrition is proper nutrition. Okay, pH is normal. Okay, still no accumulation of toxic product. Still bacteria not division. Bacteria never division, and it never accumulate any toxic material. So no accumulation toxic product. It is the one favorable condition. Okay, and O2 supply is huge number. Okay, another, no cell growth. Still they have no any competitor. Okay, so they can grow very rapidly. So here we see in graph, that time the growth is exponentially phase, very rapid growth, exponential phase. So all favorable condition is there. So that time it takes rapid growth, exponential growth. That's why the curve is look like that. Okay, like this phase is called, between this one to this one is called the log phase. Okay, this one is the log phase. Clear? Now comes to the third point, stationary phase. What will happen after rapid growth or completion of the log phase? What will happen? Nutrition, not proper nutrition. What happened? Exhaustion of nutrition. Okay, clear? Then pH. Why? Because of the rapid growth of bacteria. So it can generally release so many enzymes. So enzyme has, it may be acidic or alkaline. So it can change the media nature of the media pH. Okay, so pH change. Okay, after that, after rapid growth, bacteria will accumulate few toxic material. So accumulation of toxic product. Okay, next, O2 supply reduced. Okay, number of cell growth increase. Okay, so number of population increase. So that time what happened? So these are all are unfavorable condition. Already nutrition is decreased, exhaustion, pH change, toxic product already accumulate, and O2 supply is reduced, and number of cell growth increased in the whole media. So that time it will get unfavorable condition. That time the bacterial growth will stop. Okay, and it goes when, if you see the curve, that time we see a serial line, plateau stage. Okay, look like this. No extra growth of bacterial cell because all are unfavorable condition is present, all are, so that time bacteria growth will be hampered. So this phase is called the stationary phase. Okay, now for number 4, decline phase. What will happen in decline phase? In decline phase, it, here we see it goes towards the down in condition. But why? Because after so many population of the bacterial growth, all are exhausted, all are unfavorable condition. So exhaustion of nutrition completely exhaustion. There are no nutrition present inside the media. Okay, pH is changed totally. Okay, accumulation of toxic product, huge number. Okay, huge in number, and O2 supply is totally reduced. Okay, and number of cell increase more than, so many bacteria increases. So that time what will happen? No bacteria will grow. If some or few bacterial colony will die, that time if any bacteria get favorable condition, that time against of this one or single colony will be produced. So that time the, because of the unfavorable condition, bacterial cell will die, and that's why the graph goes down. So after that we can confirm that when, if you growth a single colony over a suitable media, after few days interval, it generally, bacteria will generally die. Okay, so we seen the decline phase. Okay, decline phase, because of the all unfavorable condition is there, the bacteria will never grow inside this. Okay, so these are total bacterial growth curve here. So when we introduce any suitable media, primarily they never grow because of why? It takes few time to adaptation of the nature. Okay, after that it grows exponential because all favorable condition is present inside this. After that, exhaustion of nutrition, pH change, accumulation of toxic product, O2 supply is reduced, number of cell growth increase. That time it will growth very limited in growth. Okay, that's why we seen a state curve. After that, totally exhaustion of or totally unfavorable condition is present inside this, the bacterial cell will die randomly, and the graph is down in lower portion. Okay, so that is the main things as per respect to time and the number of cell growth. Okay, so this one is the bacterial growth curve. Okay, so I think it is very helpful for you. Thank you.